The cell profiling module is dedicated to single-cell RNA-seq datasets. In this module, you can profile your individual cells based on known reference sets. So first of all, on the left-hand side, you have an options menu. From it, you can select whether you want to focus only on relevant sample groups, rather than including all of them in your analysis. And you can change the layout of the plots by selecting either PCA or default plots, default being TSNE plots. So first of all, we have the cell type profiling plots. These are computationally inferred using deconvolution methods against known reference set. Using the settings icon, you can choose the reference set you want to compare your samples against, and the deconvolution method that you want to use. You can also further customize your plots by sorting them by probability or name or changing the layout. On the left-hand side, you have phenotype plots. These are basically TSNE plots overlaid with the various phenotype descriptors from your original input data. And you can see, for example, for this particular plot that relates to the group phenotype, that this cluster here is described as B cells. And indeed, when we look back at the cell type profiling plots, we can see that this cluster corresponds to naive B cells. Next, we have the mapping tab. The mapping tab, first of all, produces either a dot map or a heat map plot, where you can basically see the similar information as the clustering plots before, and you can match your phenotype groups with various reference datasets. So again, you can select different reference sets using the settings icon, as well as changing analysis methods, and you can also sort the samples by the different input phenotypes. On the right-hand side, the platform produces a proportion plot. This proportion plot can be customized via the settings icon, so you can change what is represented on the X and Y axis. So in this case, I want to change the X axis to represent the group phenotype from my data. And now I leave of course the y-axis the same as this reflects the different descriptors from the reference data set. And so here you can see, for example, that for my phenotypic group B cell, the vast majority of the individual cells that make it up are mostly naive B cells and memory B cells, which is consistent with their classification. The markers tab shows the distribution of expression of marker genes with TSNE plots. So this allows you to select, first of all, you can perform this analysis at the gene or gene set level, and you can select from various feature sets that can be either gene families or gene sets if you chose those. You can also filter your outputs by, in this case, gene names or gene set names. And you get here for each one of your cells in the data set the expression levels for specific genes, in this case, or gene sets if you were to choose those. On the right-hand side, the platform produces a cytoplot. This basically mimics the scatterplots used for gating in flow cytometry analysis. And by going to the settings menu, you can change, you can select from a list of genes which one to use for the X and Y axis. Finally, we have the iTALK tab. This tab is used to profile and visualize the ligand-receptor mediated intracellular crosstalk signals from your single-cell RNA-seq data. What it does, it produces 3 plots. The first is a circle plot that visualizes the communication structure of ligand-receptor genes. You can change this plot from the scroll-down menu by selecting a different phenotype descriptor as defined in your input data, and you can select from 4 different gene categories for finding ligand-receptor pairs. Through the settings, you can also increase or decrease the number of pairs that you want represented. The expression heat map is showing the expression level and log4 changes of the ligand-receptor genes. And finally, the net view plot visualizes the communication structure of ligand-receptor genes and can again be changed by increasing the number of top genes. All these 3 plots can be changed via these menus. So, for example, if we go instead for group and we change this to growth factor, then we get a different set of plots for your data.